Imagine your brain isn't just this like vast network of firing electrical wires, but an incredibly sophisticated, hyper-local climate control system.
Right, which is a pretty huge shift from how we usually picture it.
Yeah, exactly. I mean, I want you to picture a sprawling building where every single room, every winding hallway, every tiny closet has its own custom-tailored environment. And it's specifically adjusted second by second to keep everything running perfectly.
And for decades, we just assumed these tiny environmental controllers were, well, completely static.
Right. We thought they just set the thermostat to a single unchangeable baseline and just, left it there.
Yeah.
But, well, we were wrong.
Very wrong, yeah.
So today we are taking a deep dive into a 2026 Nature Communications paper by Jan Meyer and colleagues. And our mission today is to completely dismantle that old assumption about how the brain maintains its environment.
It's a fascinating paper. We're looking at astrocytes, and we're going to see how they use a hidden mechanism involving sodium to keep your brain from essentially constantly short-circuiting.
Okay, let's unpack this. Because the crux of this entire system revolves around the sodium inside these astrocytes, right?
Yes, absolutely. The baseline assumption for a really long time was that astrocytes keep their internal sodium levels very low.
Yeah.
And uniformly low across the board.
And they use that low internal sodium as a kind of suction force, right? Like an inward gradient.
Exactly. Because the space outside the cell is salty and the inside is not, sodium desperately wants to rush in.
So the astrocyte uses that rushing force to pull in extra potassium and spilled neurotransmitters like glutamate from the space around the neurons.
Yeah, that inward rush is critical. I mean, without that gradient, the brain's extracellular space would just quickly become toxic.
Right.
But to understand what this new research discovered, we first have to look at the limitations of what we, used to think. Earlier studies relied almost entirely on bulk measurement.
Meaning they just measured the whole thing at once?
Pretty much. Scientists would look at the main cell body of the astrocyte, take an average reading, and that average suggested a uniformly low sodium concentration everywhere.
Which I guess makes logical sense. Like, if a low sodium baseline is required to power all this vital cleanup work, you'd assume the cell vigorously protects that low baseline in every single cell.
Exactly. It made sense based on the tools we had.
Let's use a visual to lock in the mechanics of how that cleanup actually works. Think of the astrocyte as a highly advanced cellular vacuum cleaner.
I like that analogy.
Thanks. So that low intracellular sodium we just mentioned, that is your suction power. The astrocyte uses this suction to pull in the dirt.
Which, in this case, is the spilled glutamate from all that neuronal firing.
Right, and it pulls it through specialized intake valves called EATs, or excitatory amino acid transporters. But obviously, if you're constantly sucking in sodium along with the glutamate, your vacuum is going to fill up.
Yeah, you completely lose your suction power.
So the astrocyte has a motor running constantly to pump that sodium back out. And that motor is a protein pump called the NKA, or Na plus K plus ATPase for short.
The vacuum concept holds up mechanistically, for sure. But we need to look at the flaw in how we previously measured its power.
Right, those bulk measurements.
Yeah, those measurements were fundamentally limited by the tools available at the time. This team of researchers, Jan Meyer and the group, utilized a breakthrough optical tool to get past those limitations.
And what was that?
They used MP-FLIM, which stands for multi-photon fluorescence lifetime imaging microscopy.
That is a mouthful.
It really is. And they combined this with something called time-correlated single-photon counting, or TCSPC.
Okay, wow.
Using a highly specific chemical dye known as ING2.
Now wait a minute, I have to push back here. Why couldn't scientists just use standard glowing dyes? I mean, we've been making cells glow under microscopes for decades to see what's inside them.
That's true, we have.
So if the dye binds to sodium and glows, shouldn't we be able to just, look at how bright the cell is to see how salty it is? Why do we need lifetime imaging?
Well, what's fascinating here is that standard intensity-based chemical dyes are incredibly prone to optical artifacts.
Artifacts, like errors.
Exactly. Think about the physical reality of a microscopic cell. If a particular spot under the microscope glows brighter, is it because there's actually more sodium in that spot, or is it just because more of your chemical dye accidentally pooled up in that part of the cell?
Oh, I see. Or maybe the tissue is just slightly thicker there.
Right. You just can't know for sure. So FLIM bypasses that entirely. It doesn't measure how bright the dye is. It measures the lifetime of the photon.
Wait, the lifetime? Like how long the light lasts?
Yes. We are talking about literal nanoseconds here. Imagine you're not judging a fire by how bright the flames are, but you have a microscopic stopwatch timing exactly how long the afterglow of a single photon lasts before it completely fades away.
So the speed of the fade tells you what's in the room.
Precisely. This decay follows a very predictable mathematical rule called a Michaelis-Menten relationship. It's essentially a speed limit for the chemical reaction.
Okay, so how does that work with the dye?
In this specific brain tissue, the dye has an apparent KD of 21 millimolar. That's a biophysical term, meaning that right around the 21 millimolar mark.
Which is the normal range for these cells, right?
Exactly, the physiological sweet spot. At that mark, the dye's fade time is exquisitely sensitive to the exact concentration of sodium. It is completely unbiased by how much dye is present.
That is wild.
Yeah, whether you have 10 dye molecules or 1,000, the lifespan of the photon they emit decays at the exact same rate based solely on the surrounding sodium. It gives us an absolute artifact-free measurement.
So you're literally timing the lifespan of individual packets of light to map out the environment.
That's exactly what they did.
Okay, so to recap section 1 really quickly for you listening, bullet 1. Astrocytes rely on low internal sodium to power the cleanup of the brain's extracellular space.
Yeah.
Bullet 2. Traditional measurements miss the nuance, but this MP-FLIM tool measures the lifespan of a photon to give exact, unbiased sodium readings.
Perfect summary.
So here's a question for you to think about. Have you ever considered how difficult it is to measure the exact saltiness of a single microscopic cell without totally disrupting it? It makes you wonder how many of our old assumptions in biology are just artifacts of the tools we were using.
A significant amount, honestly, as this paper proves. So armed with this unbiased FLIM technology, the researchers looked at mouse brain slices, specifically the hippocampus and the cortex.
And what did they find?
Well, at first glance, the data actually seemed to confirm the old dogma. In the main cell bodies, the soma, they found the average sodium was around 13.8 millimolar in the hippocampus and 12.5 in the cortex. They even cross-validated this against an older, reliable dye called SBFI, which gave them 13.1 millimolar.
So the overall average matched the historic assumptions perfectly.
It did. But the average was completely hiding the reality.
Right, because when they mapped out all these individual cells, the sodium levels didn't just form a normal bell curve around that 13 millimolar mark.
No, the data revealed a striking bimodal distribution, a double-peaked graph.
A double peak.
Yeah. Instead of a uniform population, most cells clustered tightly around either a distinctly low peak of 8.7 millimolar or a distinctly high peak of 17.0 millimolar.
Oh, wow.
The astrocytes were physically dividing themselves into two completely distinct functional camps.
To put that in perspective, it's like looking at a massive neighborhood and reading a report that says the average home temperature is a pleasant 70 degrees.
Right.
But then when you actually walk down the street and check the thermostats, there are absolutely zero houses set to 70. Half the houses are freezing cold at 60 degrees, and the other half are sweltering at 80 degrees.
That's a great way to look at it. You have a dedicated cold group and a dedicated warm group, but the average completely obscured that reality.
But they didn't stop there, right?
No, we can take that a step further. The researchers didn't just stop at brain slices. I mean, a slice of brain tissue in a petri dish is essentially at rest.
Yeah, it's not exactly doing much thinking.
Exactly. So to see what these cells do under pressure, they used the FLIM technology on live anesthetized breathing mice. They measured the astrocytes in vivo.
And the numbers completely shifted again.
Yeah.
In the live mice, the estimated average sodium was significantly higher. It was 19.8 millimolar across the board, with this massive wild range shooting all the way up to 42.1 millimolar in some areas.
Which is huge.
But wait, why is a live brain so much saltier than a perfectly healthy brain slice? Is the slice broken?
Not broken, no. It just comes down to the environmental workload. A brain slice in artificial fluid is largely devoid of spontaneous network-level sensory input. It's very quiet.
Right, it's basically offline.
But in a living, breathing animal, there is relentless background neuronal activity. The neural networks are constantly firing to process the animal's breath, its heartbeat, the sensory inputs from the room.
Oh, and remember the vacuum analogy. More neuronal firing means more glutamate is constantly being released into the synapses.
Exactly, the mechanism. So the EAT intake valves on the astrocytes are just constantly sucking in that glutamate to keep the live brain clean.
Which brings a flood of sodium right along with it.
Precisely. The live brain is a much higher demand environment, so it requires constant, aggressive vacuuming.
Okay, let's do another quick recap. Bullet 1. Astrocyte cell bodies average around 13 to 14 millimolar of sodium in slices, but they actually fall into two distinct camps, high and low, bullet 2. In live brains, baseline sodium is roughly 6 millimolar higher because the neural networks are actively firing.
Spot on.
So my question for you listening is, what environmental factors in your own life dictate whether your body functions at a high or low baseline? Because clearly, these astrocytes are constantly adapting to the mess around them.
And how they adapt locally brings us to what might be, well, the most visually stunning part of this research. The investigators didn't just look at the main cell body.
Right, they zoomed in.
Yeah, astrocytes have these incredibly intricate, delicate branching processes that reach out from the soma, like the dense root system of a tree. And they physically wrap around millions of individual neuronal synapses.
That's incredible.
So by using a tiny glass pipette, a patch clamp, they loaded the FLIM dye specifically into individual cells so they could map out the internal environment of those tiny branches.
And the numbers they found in the branches defied the old uniform baseline completely. The branches had a significantly higher average sodium of 17.3 millimolar.
Right, much higher than the soma.
But the craziest part is the spatial gradient they discovered. The further you travel away from the cell body, down the microscopic branch, the saltier it gets.
It's really remarkable.
At 5 to 15 micrometers away, the sodium sits at 14.9 millimolar. But when you get out to the far tips, 15 to 25 micrometers away, it spikes to 20.8 millimolar.
And the variance between two different branches of the exact same cell could be up to 25 millimolar.
That is insane, within the same cell.
Exactly. Now, to prove the biological mechanism driving that massive gradient, they ran two very elegant functional tests. First, they applied a pharmacological agent called TFB-TBOA.
And what does that do?
This drug specifically blocks the EAT glutamate transporters on the astrocyte. It essentially shuts off the vacuum intake.
Okay, so what happened?
The result was that nearby neurons started to swell and overload with sodium because their glutamate wasn't being cleared. But inside the astrocyte, the internal sodium actually dropped by about 4.6 millimolar.
Because the intake was shut off. No new sodium was being pulled in from the firing neurons.
Precisely. Then they ran the inverse test. They flooded the surrounding tissue with a high potassium solution, 10 millimolar of potassium.
Because astrocytes also have to clear excess potassium to prevent seizures, right?
Yes, exactly. So this high potassium load forced the astrocyte's NKA motor, the pump, to shift into absolute overdrive to clear it.
Oh, I see where this is going.
Because the NKA pump operates by swapping potassium for sodium, physically pushing sodium out of the cell to bring potassium in, the astrocyte's internal sodium plummeted down to 6.3 millimolar.
Wow. Okay, let's build a mental model for this, because visualizing this gradient is totally crucial. Let's look at this whole astrocytic cell as a massive river delta.
Oh, I love that imagery.
The main cell body, the soma, is the calm, vast ocean, where the overall sodium concentration is relatively low. The tiny tips of the branches, reaching far out to wrap around the firing neurons, are the turbulent, narrow rivers high up in the mountains.
Right, where all the action is.
Exactly. So every time a neuron fires, it creates a localized storm. Those EAAT intakes suck the rain, the sodium and glutamate, into the narrow river tips. Because the volume is so small and the intake is so high, the tips become highly concentrated with sodium.
It builds up fast.
But physics dictates that things naturally flow from high concentration to low concentration. So the sodium flows passively down the branch rivers, away from the turbulent synapses, and dumps into the vast ocean of the cell body.
If we connect this to the bigger picture, it is an absolute marvel of thermodynamic bioengineering. Those branch tips are on the biological front lines, intimately wrapped around the synapses, and packed with transporters constantly pulling in sodium.
Right.
But if they had to pump all that sodium back out locally, right at the delicate tips, they would burn through the cell's entire ATC energy reserve almost instantly.
They would literally exhaust themselves trying to run a heavy motor in a tiny space. But if the tips are constantly taking in sodium, why don't they run out of energy?
That is the genius of the gradient. Because of the river delta effect you just described, they don't have to pump it out locally. The sodium naturally and passively flows down the gradient to the massive soma.
Oh, wow.
The astrocyte outsources the heavy lifting. It allows the high-volume cell body, which has abundant energy resources, to handle the bulk of the NKA pumping without exhausting the local branches.
It's an incredibly elegant, energy-saving diffusion mechanism. So, recap number 3. Bullet 1. Sodium levels aren't just different between cells, they vary wildly within a single cell's branches. Bullet 2. Sodium is highest at the tips near synapses and flows downward toward the cell body.
In bullet 3, that gradient acts as an energy-saving diffusion mechanism.
Exactly. So, a question for you. Imagine having entirely different metabolic zones in your hands versus your torso. How would that completely change your efficiency?
It would be incredible. But to understand how the astrocyte actually builds these different metabolic zones, we have to look at the molecular blueprints.
Right. How do you get that bimodal distribution? How do you get half the houses in the neighborhood permanently set to cold and half to warm?
It comes down to the genetic hardware, specifically the subunits that make up the NKA pump.
Right. The pump isn't just one static piece of machinery, is it? It's made of interchangeable protein parts. Astrocytes broadly express what's called the alpha-2 subunit, but they can combine it with either a beta-1 or a beta-2 subunit.
Exactly. And using techniques like RNA-scope to look at the spatial genetics, the researchers found that 10 to 30% of the cells combine the pump with a beta-1 subunit.
And that subtle protein swap changes everything, doesn't it?
It really does. The alpha-2 beta-1 combination creates a motor with a very high affinity for sodium. That means it binds and pumps out sodium very aggressively, even when the internal concentrations are low.
So it works overtime to keep the baseline sodium firmly suppressed.
Yes. The rest of the cells, however, utilize the beta-2 combination, which has a much lower affinity. It's less aggressive, allowing for a much higher baseline of sodium to build up before it really kicks into gear to clear it out.
So the physical hardware actually dictates the baseline.
And the researchers actually proved this by running advanced biophysical computer simulations. They built a virtual astrocyte model and mixed these variable subunit compositions.
Well, also varying the expression levels, right, from 60% up to 180%.
Exactly. They let the math run, and the computer simulation perfectly replicated the double-peaked bimodal distribution they saw in the live mice.
That is so cool.
Furthermore, when they programmed the simulation to concentrate more EAT transporters and beta-2 subunits specifically at the far branch tips, the computer autonomously generated the exact river-delta diffusion gradient they observed under the microscope.
Here's where it gets really interesting, though. Astrocytes don't operate as lone wolves. They don't manage all this stress and isolation. They are physically hooked together by gap junctions into a massive interconnected cellular grid called a syncytium.
They literally share their internal cytoplasm through tiny tunnels.
Right, and this is a critical piece of the puzzle. The researchers applied a drug called carbinoxolone, or CBX.
Which chemically blocks those gap junctions.
Exactly. It forcefully severs the connections, forcing every single astrocyte to unplug from the grid and operate completely alone. And the local climate control system basically collapsed.
It really did.
When the network was blocked by CBX, the overall average sodium spiked from 13 up to 20 millimolar. But the variance is what's truly shocking. One single isolated cell's sodium skyrocketed to an astonishing 96.6 millimolar.
This raises an important question, What does that catastrophic failure tell us about the purpose of the network?
It proves they need each other.
Yes. It proves that without the physical grid to share the thermodynamic load, individual astrocytes are quickly and easily overwhelmed by localized neuronal firing. The data even showed a distance correlation.
What do you mean?
Astrocytes that are physically located further away from their neighbors exhibit much higher sodium variance than those tightly clustered together. They are not isolated independent units.
They function as a shared, highly cooperative community that dynamically tailors itself to the exact demands of the local neural network.
Perfectly said.
All right, final recap. Bullet 1. The two distinct camps of astrocytes are driven by different protein engines, the beta-1 versus beta-2 NKA subunits. Bullet 2. Astrocytes use gap junctions to act as a communal grid, sharing the burden of sodium processing.
Excellent.
So if astrocytes share their stress with their neighbors, how might a single localized brain injury cascade through that invisible glial network?
That's a profound thought. If we pull back and look at the ultimate takeaway of this immense body of data, it is a total redefinition of the astrocyte.
Right. They aren't just passive glue.
Not at all. They possess profound cellular and subcellular heterogeneity. Those high sodium compartments, the ones utilizing the beta-2 engines at the tips of their branches, are specifically geared toward aggressively clearing neurotransmitters.
They are physically altering their own internal physics to control the excitability of the exact specific neurons they are wrapped around. We have journeyed from dismantling an old myth based on bulk averages to using the lifespan of single photons to map a cell, all the way down to the interchangeable protein motors inside a microscopic branch.
We've seen how the river delta flows and how the neighborhood shares its power.
So what does this all mean for you? Well, I want to leave you with a thought that builds on everything we've just uncovered today. We've always been taught that memory and learning are strictly matters of neuronal wiring. Neurons firing together and wiring together.
Right, that's the classic model.
But if astrocytes physically alter their internal sodium machinery based on the specific neurons they surround,
Which we now know they do.
right, if they act as a hyperlocal climate control system that permanently adapts to the local weather, could our very memories, our habits, our deeply learned behaviors be stored outside the neurons?
Oh, wow.
Could the actual muscle memory of how to ride a bike or the emotional resonance of your favorite song be partially encoded in the highly customized long-term sodium tuning of the astrocytes wrapped around those networks?
That is a profound implication. It shifts the entire paradigm of how we understand the biological basis of learning and memory.
It really does. So the next time you find yourself focusing intensely on a difficult task or learning a new skill, just picture those millions of tiny river deltas in your brain, silently adjusting their flow, perfectly balancing the storm. Thank you for joining us on this deep dive into the source material. Keep wondering, keep exploring, and we'll see you next time.